Okay, thanks very much, Davor. Hello everyone. It's a bit unnerving, I have to say, still getting used to the idea of talking to the screen of your laptop without any faces that you're actually looking at while speaking. So the topic that I've chosen, and thank you very much for the invitation to and the opportunity to speak. I'm talking about chromatin wiring and rewiring in the heart and heart failure. So the basis for all of this work really comes from the idea that gene expression changes are very fundamental and underpinning for a lot of the processes we see in the heart, especially when it fails. So just going to turn my pointer on this screen here. So regardless of what the original cause of the heart failure is, be that ischemia, metabolic disease, hypertension, and so on, you will always find recurring features that unify all of those different original causes. And then characteristically, what you also find is that the signals going across from the cell surface right down to the nucleus converge down on gene expression changes. And for these gene expression changes, we're all very familiar with the full spectrum of this hallmark of gene expression changes. So sticking on gene expression changes too, if you look at how genes then change as a program going from comparing between healthy hearts and heart failing hearts, you see a classical display of those gene expression changes, as we said. And what's been shown, papers from quite a while ago, is when you've treated patients with left ventricular assist devices or even beta blockade agents, there is that propensity for gene expression changes to reverse, which suggests and speaks to the plasticity of this phenomenon. And the best part as well for us in basic research is there is a really good model for such gene expression changes as well in the form of the models such as transverse aortic constriction, which, as we all know, is a pressure overload model for heart failure. So if you are like us that likes to think about epigenetics arising from this, then it becomes as if we have an epigenetic-centric hypothesis for heart failure, which is really to say that epigenetics integrates all of these myriad causes, upstream causes that can give you heart failure, and unifies all of them to produce that single heart failure phenotype. And if that was the case, then chromatin modeling would be something of great interest to figure out. And potentially, if we find means to get to it, you could be getting down to novel therapeutic approaches for heart disease. But actually that concept is not new, obviously. So some really classical and good examples of papers going quite far back that showed us that the histone deacetylases were very central, playing a role in these gene expression changes that we see. And ever since then, even more pieces of evidence coming through with readers of histone acetylation or histone deacetylases. And then more recently, even going to histone methylation, which is in this case a eraser of histone methylation or a writer of histone methylases. So the part that our lab got interested in was really not at the methylation changes at different loci in the genome or the histone changes at different locations, but more a question of how the chromatin is folded or differentially folded if it was the case in controlling gene expression. So for a time up to then, it's become possible using technology to study how chromatin folds. And a few principles that I wanted to lay out before getting down to the data was how it began to be discovered that the folding of chromatin is in a very organized fashion. It's not random, and it's so-called a fractal globule model. it also starts to explain how enhancers picks its corresponding gene to be interacting with, resulting in the explanation for these things known as some people call enhancer conundrums. Why does an enhancer interact with one gene upstream to it and not the downstream gene, or vice versa? And how does this get controlled? And it seems chromatin folding is giving an answer to some of that explanation. And then another model of the understanding chromatin folding by way of compartmentalizing chromatin or genomic locations into these things called topologically associated domains. And really domains are being made up of so-called insulated neighborhoods. So an insulated neighborhood, as a matter of structure, is a situation where genes within an insulated neighborhood are in a context where they are more likely to interact with each other, the enhancers within the neighborhood with genes within the neighborhood. And it turns out that the proteins that bind to DNA at the anchors of these insulated neighborhoods are now known to be proteins such as the CTCF protein, which is a protein that we will be talking a bit more about, and also cohesin ring complex proteins. So again, these are now protein complexes that we've begun to understand a lot more about, and fundamentally playing important roles in the way that chromatin is folded. So more on the principles of chromatin folding. DNA loops are brought from distal regions into proximity as a result of the folding, as you see here. Enhancers and promoters interact within the loops and within these, as we say, insulated neighborhoods. And again, the point, CTCF appears to sit at the anchor of these loops and hold as a scaffold holding these loops together. So more than just that, it's also been quite important to study enhancer locations across the genome. And why is that? Because it turns out that enhancers, as marked by DNA's hypersensitive sites in this occasion, are the location in the genome where a lot of the phenotype SNPs, phenotype disease-related SNPs found in GWAS are quite often located in enhancers as well. And when enhancers, and this is one good example of a very notable paper, when enhancers are brought into proximity with very distally located genes far away from each other, then it begins to explain how enhancers within a position such as our chromosome 9p21, very famous locus for coronary artery disease, is associated with this distal gene. And it's really the SNP in this position of the enhancer as a result of interacting with this distal gene, which is interferon gene in this occasion, ends up controlling expression of the interferon gene because the enhancer SNP change affects whether a transcription factor, in this case STAT1, whether it binds or not bind, affecting the expression of the distally located gene interferon. So again, the point to make that genetic enhancers in enhancer sites, genetic variants in enhancer sites are worth studying because they really could give an explanation for why these genetic variants are important in disease contexts. And if we know which genes they interact with, it then gives the insight to the mechanism for how those variants are functioning. A couple of last points before we actually go on to the data. This point to make where it's also becoming quite reproducibly understood that the way that chromatin is folded into a loop, as we say, is down to the anchors where in the first place, a chromatin is extruded into the cohesin complex, ring complex, this one here. And what then occurs as these pieces of biological data shows is the chromatin then extrudes, the loop extrudes through this ring complex. and it stalls and stops extruding when it meets up with two convergent CTCF proteins, one binding on either side of the two anchors of the loop. So this is the so-called chromatin loop extrusion theory, which is believed to be the reason how gene neighborhoods are set up. So the project aims, when we were understanding all of this biology, we crafted these aims up for what we wanted to study. We wanted to generate a map for how the cardiac chromatin is organized, wanted to understand interactions between genes and enhancers, be they close together or more likely and more importantly, if they were very far apart, to understand that relationship. We wanted to use then a model where we took CTCF knockout as a model to understand how chromatin is organized in the heart. And then to ask the question whether in heart failure progression, are there any changes? Do they then occur in chromatin loop changes or gene neighborhood changes? This is really taking from the point where, you know, if as clinicians we talk about things like left ventricular remodeling, we wanted to ask the questions, does the chromatin remodel as well? And then, of course, how does the gene neighborhoods then impact upon these classical hallmarks of gene expression change that I showed very early on in the first couple of slides. So we took the CTCF conditional knockout mouse, and the model that we chose to induce the knockout was to inject AAV9 Cre recombinase viruses that were driven by CTNT promoter. So as you can see here, it produced quite a good degree of knockout when we ran Western blots on the purified cardiomyocytes from these mice, and reproducibly or consistently also showed that both at the histological sections levels and also qPCR levels, CTCF was properly knocked out. So just by means of introduction, Dom is now a postdoc. He's finished his piece of PhD work. And George was the green fingers that could do a lot of the high C, 3C distal interactions assays that was involved in this work. And Wilson's our inimitable bioinformatician for this piece of work. So we were really feeling very lucky when we saw that indeed the CTCF knockout mouse developed consistent dilatation of the heart. This was a phenomenon that we reproduced very neatly. But what was more important, we thought, was trying to figure out, as we say, the basis and understanding for what was going on when CTCF was depleted. And right around the time, there was quite a number of conflicting reports appearing over the question of what was happening to the loops, the insulated loops, upon CTCF depletion. For example, in a paper published here, you can see that loops did not appear to be dissolving or disappearing upon CTCF knockout. So a bit more explanation of how to interpret this figure here. This figure is generated by a so-called Hi-C method, which is a 3 chromatin conformation methodology to map out distal genomic interactions. And the way to interpret this really is on the x-axis and the y-axis, you have the genomic position. So A and A genomic position are obviously going to highly interact with each other, hence the dense red color. So intensity of the red is the number of reads that you get from distal interactions. At the point where you see a high spot of red, highly intense spot of red, this makes you then conclude that genomic location B also interacts with genomic location A. Whereas in the position where there isn't a spot of red, location C does not interact with location A. So as you say here, there was the discovery or the findings reported in the literature that despite CTCF knockout, the loops still seem to be present. And this was yet another paper that suggested in aggregate, loops did not seem to disappear until a paper from Benoit Bruno's lab then showed us that in his model, when CTCF was depleted, using an auxin degron methodology of degrading CTCF, there was the disappearance of loops. So up until that point, it was still so these were measured in aggregate, and up till that point, it was still a question of whether, how much did loops disappear when CTCF really was knocked out. So we took our data and analyzed it carefully, and what we really came up with, collaborating with Ares, who is one of the key leaders in describing how loops can be computationally derived and calculated, we discovered that up to 99% of loops really are lost in our CTCF model when we took out purified cardiomyocytes from these hearts to examine them. And then we did the further analysis, which going forward from if loops were lost, so if these CTCF loop boundaries were lost, what then happened for the interaction of genes and enhancers within loops. So as you can see here, the bulk of interactions occur within insulated neighborhoods of CTCF, as I said here. And when we took out the CTCF interaction or the CTCF loops, when the loops were taken away, the interaction started to bleed across these insulated positions. So bleed through with transcriptomic changes across chromatin loops. This is not an easy slide to explain in just half a minute, but what we managed to show as well with our model was that in the positions where loops have been taken apart, genes inside the loops could be either up or down regulated because they lost interactions with enhancers within the loops, but they could also gain interactions with enhancers outside the loops. But more interestingly, genes at the flanks of the loops were consistently upregulated, suggesting that they were gaining interactions more than losing them. So when we look back at the dilated hearts that we got out of the CTCF knockout model, what we discovered was the gene expression changes. So these are all gene expression changes of the knockout mice. We found that they didn't exactly mirror the gene expression changes of our classical sham and transverse aortic constriction mice. So there were obvious gene expression changes in transverse aortic constriction, which did not reproduce and get found in the CTCF knockout model. And vice versa, there was gene expression changes in the CTCF knockout model, which were not replicated in the TAC model. So this was speaking to the biology that these two mice were getting dilatation from quite different backgrounds of gene expression changes, although there were consistent expression changes as well, we noted. So we honed in on a particular locus because this one really caught our attention. So MPPA and MPPB, as everyone well knows, they are hallmark gene expression changes markers that gets upregulated at least in a wild type mouse when there is myocardial stress as a myocardial stress response. But what we noticed in our knockout mouse model, the upregulation of MPPB was consistent, but MPPA was not upregulated, and it wasn't upregulated even when we performed transverse aortic constriction on the knockout mouse. This then focused our attention down to both of these genes, and we reminded ourselves that these two genes were actually sitting next to each other on a genomic locus, and in fact they sat within a CTCF loop, as shown in this cartoon here. And what was interesting, and of course we demonstrated that upon CTCF knockout, the loop was taken apart, so the loop was lost. And to get to understanding what was going on in terms of the interactions at this locus, we undertook this procedure called 4C sequencing, which is really to ask from this locus, taking this anchor of a locus, what else was interacting with this locus in this location here, in this position here. And we found that within the CTCF loop, indeed, there was an enhancer upstream of MPPA in this case that was interacting with MPPA, but that interaction is lost in a CTCF knockout location, in a CTCF knockout cardiomyocyte. which to an extent then explains why NPPA is unable to get upregulated in the CTCF knockout mouse. And then actually also equally interesting when we did the 4C-seq on the NPPB locus. So we can see that NPP locus also interacts with this same enhancer that is upstream to it. So NPPA and NPPB share the same enhancer within the CTCF loop. And interestingly, when we, and in the tag situation, we can see the enhancers are interacting as expected. But in the CTCF knockout mouse, when the loops have been dissolved or dissipated, what we saw was a bleed through with an interaction to an enhancer that is even more upstream, which usually is not present as an interaction in the wild type setting. So this was the model we ended up putting down in a picture, in a cartoon to describe what happens. Here's your insulated CTCF loop, anchors of the CTCF and the cohesin complex. And under normal circumstances, the upstream enhancer is within reach to the two NPP and NPPB gene, but upon the dissipation or disappearance of the CTCF loop, NPPB is still able to interact with the upstream enhancer, but not NPPA. And because the loop has been taken apart, it's now able to interact with an enhancer that used to be outside of the insulator loop. So the summary here at this point, loops fall apart in CTCF knockout mouse. And what we think we are seeing is the ectopic enhancer promoter interactions bleed across the insulation when loops are broken, giving rise to ectopic transcription. And we believe that the mechanisms for dilatation, are not the same, or mechanism is not the same between CTCF knockout and TAC. But what then happens in a TAC setting? So this was still the question that we wanted to get to. So this is when we went back to the TAC mouse to explore what happens to CTCF loops. And really we saw that, the large abundance of CTCF loops remain very much the same. They're not changing as they do change and disappear in a CTCF knockout setting. But what is changing, on the other hand, is really that the enhancer enrichment for histone 3 lysine 27 acetylation, so K27 lysine 27 acetylation mark changes are the ones that are correlating well with gene expression changes, either when they're downregulated, they become reduced in the enrichment of acetylation, or when they get upregulated, they are more enriched with acetylation. And we managed to build up a series of networks of genes based on their interaction between same enhancer families, and showed that genes that are in the same enhancer family tend to get upregulated together, and genes that are downregulated tend to get downregulated together as these groups of enhancer promoter interactions go. So CTCF-based, the summary we made at this point was that CTCF-based chromatin architecture seemed unchanged at least in the TAC model that we looked at. And instead what was changing were enhancer of K27 acetylation enrichment that was changing. So, one more summary at this point. We managed to build up, let's say, the CTCF framework and show that the CTCF framework of architecture is largely stable during attack myocardial stress response. And if the enhancers were the ones that were changing, then it really would be worth mapping out which enhancers are connecting with which genes. So since then, our lab's gone on to work with human hearts now, looking at the interactions of distal genes with their enhancers in very long distances. So it's not specifically written on here, but you can see that the interaction is typified by intense red spot as shown here. And instead of simply, so this is actually an enhancer. We have to take a really zoom in view. It's not just next to the gene NOS1AP, but it's an enhancer that's situated a little bit upstream of NOS1-AP. But you might think that this enhancer interacts only with or has regulatory effect only on the nearby gene NOS1-AP. But in fact, it also has interactions with a very distal gene, which in this case is C1ORF226, and absolutely no detectable interactions with a gene just next door to it, but upstream, or ORF ML2B. So not shown here, we have since shown that the interaction is also corresponding to gene expression changes of this gene when enhancer enrichment is also changing. Whereas with a gene that doesn't interact with, the gene expression changes of that gene is unrelated to the enhancer mark. So more examples of genes that we might be more interested in. So in this case, ACTC1 gene, we found that this gene, for example, has 3 enhancers that it interacts with. And we used a algorithm ABC scoring system and discovered that this is the key enhancer in this case for ACTC gene. So a conclusion that we can draw at this point is that enhancers really can skip over genes, as you can see in this setting here. And in quite a lot of these cases, the distal enhancers can be many hundreds of kilobases away. And quite frequently, we're finding that obviously there's more than 1 enhancer per gene. And in quite a number of cases, as we saw in the last one, some enhancers interact with more than 1 gene as well. So in the case of Actor1, which is here, we took the main enhancer that we concluded to be based on the ABC scoring system, as I said, and we deleted the enhancer using CRISPR and showed that upon CRISPR-mediated deletion of the enhancer, it actually does downregulate ACTC1 gene expression. And the same for MYL2, where the distal enhancer that we annotated was in this position. When we deleted it, the expression of the gene also nicely got downregulated. So I think I'm nearly at the end of the slides. My conclusion at this point, not just that the CTCF overall structure is largely stable. We see that histone 3 lysine 27 acetylation changes associate well with gene expression changes. And as a result, we have really gone deep into map enhancer, promoter, and gene interactions with each other. So what next? What's possible applications for such enhancer gene maps? If we wanted to then look at genetic variants within distal enhancers, this sets the stage nicely to do so. And going forward, it might be possible to regulate gene expression and control them, in experimental models, potentially by targeting enhancers, for example, using the system of Cas9 fused with epigenetic modifiers to target for gene expression control rather than targeting the gene directly itself. So lots of this work was done by teamwork. So I pointed out Dom is no longer in the lab. He's a postdoc now in another lab in NUS. And George, I mentioned, there's also Tuan, also worth mentioning. He's the animal, together with Peter, our animal specialist in the lab with surgery and echocardiography and so on. And also to mention Wilson, whom I'm trying to pick up from here. Here he is, who is our bioinformatician, who's done plenty of bioinformatics work beyond just the one that you've heard. And also note of thanks to our collaborators that helped us pull this work together. So in this stressful times of COVID, I have to say all the lights are not turned on in Singapore at this point, even though it's evening. We're in a severe lockdown situation as well, as many people know. But this is where our lab is, in the A-Star campus, our hospital and university. And I put a shout out here to say that our research program is on the lookout for group leaders, postdocs, and PhD students. And at this point, I say thank you and hand the microphone back to Davor.
Well, Roger, thank you very much. And I mean, obviously, to everybody who's tuned in, obviously, Roger's hiring, so amazing lab. I encourage everybody, I've been there several times, I would encourage everyone to join his lab. So it's, and Singapore is beautiful, even when the lights are switched off, by the way. And I'm sure this will pass. So, I mean, Roger, first of all, thank you for, obviously some of you know already, but it's past midnight in Singapore. And Roger's our first speaker that's not from North America or Europe. And so thank you for staying up so late to give us an amazing webinar, and also for disclosing so much of non-published data as well, which is something that a lot of people are doing and amazes me. But clearly this is quite a nice forum where people are very happy to share, and hopefully it's going to get published soon. So please feel free to use the Q&A box for questions. And I leave it up to Roger now to go through them at your own leisure, Roger. So.
Okay, so I think there are, I can start from the top, I think that's the best thing to do. So first question was the question that asked can everyone else see the questions? Q&A, Davor?
I think yes. I think everybody should be able to see the questions.
Okay, so the question was
still helpful at least to read out some parts of it. It's
whether we could group the disrupted gene spatial interactions to certain classes of genes. So interestingly, there are genes, clusters of genes that are located at one location, such as MPP and MPPB, that you could say has a reasonable relationship with each other. So in those cases, quite frequently, they do share spatial interactions and share enhancers, whether that's the direct answer to that question. But actually, on the other hand, quite frequently, we're still trying to figure out why certain genes share enhancers when it doesn't look like they have any specific reason to be related to each other. Next question is CTCF gene specific to the heart? No, absolutely not. So it's ubiquitously expressed. It was to such an extent of ubiquitous expression that people asked the question, why was I deleting a ubiquitous gene in the heart? But really it was just to get down to a model to be able to understand and study chromatin interactions, as I said. So no, it's not a gene specific to the heart. Next question was, what will be the effect of mutation on CTCF gene? Will there be any damage to loop formation? So yes, absolutely. So in fact, CTCF mutations are found in human patients. So these would be individuals that have, it's a syndrome and they have this is a recessive condition. And the loss of CTCF function produces, as a phenotype, it's mostly neuropsychiatric disorders, developmental disorders. And I don't think I've seen anyone take iPS cells from these individuals to study how the loop formation is damaged, but that would be an interesting thing to do. We have one CTCF individual knockout, or not knockout, CTCF missense individual in Singapore as a de novo mutation that the Children's Hospital is looking after. Okay, so next question was, this is a question that's a bit fragmented and I'm not sure what it actually refers to, so I might skip that one. What will be the role of this gene in hypoxia ischemic conditions? So we actually didn't see any big changes in CTCF in all the different kinds of stimulations that one would normally use to stimulate changes on cardiomyocytes. So for example, you might be wondering if, say, hypoxia or inflammatory or ischemic conditions or even hypertrophic stresses on the cardiomyocyte, does it change levels of CTCF or does it induce post-transcriptional changes of CTCF? No, we haven't found any of those. So CTCF really seems quite stable, and the insulated neighborhoods also seem very stable. Here's the question. Correction, H3K27AC seems absent in the MPPA locus. any explanation of this marker in relation to expression levels of MPP-MP? So we find that the myocardial stress response, at least generated by the transverse aortic constriction, really does require this loop to be present so that the MPPA gene and MPPB gene can engage this distal enhancer and give rise to their upregulation in a myocardial stress response. And if the loop was not present, then MPPB is still near enough to the enhancer to get upregulated in stress, but MPPA is silent to that now. So next question was, is CTCF, this was asked by Delphine Mika. Hello, Delphine. Is CTCF also deleted in the sinus node? Actually, this is a good question. I don't know if we have the expertise in our lab, despite such a number of us, to isolate the sinus node of the mouse. It might be a little bit challenging. I have to go back and scratch my head and think about that. So we don't know. I might be needed to also consider whether using a CT&T promoter driving the Cre recombinase, whether that would delete CTCF in the sinus node. Maybe if troponin's expressed in the sinus node, then maybe. Any evidence of CTCF involvement in heart failure? I would have to say at this point, no, because for example, GWAS studies for heart failure is not producing any hits, genetic hits around the CTCF gene locus. As I said, CTCF levels are not seemingly changing in human heart failure. So where we really are is just having been able to use this as a means to get into studying and understanding the chromatin organization, rather than saying that it's a target for heart failure in disease. So no obvious involvement in heart failure as such now. So Jessica Filger asked the question, do different types of genes have different numbers of enhancer interactions? So we haven't taken families of genes to study as such. Maybe we can do that now. But what we are clearly seeing is that there are some genes that have as many as 10 enhancers that they interact with. There's nothing obvious that makes us think and realize why they're so special to have 10 enhancers, whereas other genes don't. It may be a matter of how deep we have sequenced our samples to be able to pick up some enhancers having more interactions than others. Actually, I haven't got a good answer for that. Did you measure cardiac function parameters in, so we went as far as doing echocardiogram and saw the thinning of the walls and poor contractility. And we didn't really go very far beyond that. What is the cardiac phenotype? Delphine's also asking that question. I don't know if you have something more specific to ask me, go ahead. But we didn't do more than the echo that you saw and histology. There was no apoptosis markers found in the CTCF knockout mouse. What's the benefit targeting the enhancer region over the gene itself or the promoter? This is a good question, Andrew. So I think at this moment it's if you wanted to, okay, so if we are targeting so if you target the transcript, then that's basically using RNAi methodology or something similar. But if you wanted to target the template, then if you use CRISPR at the gene locus itself or the coding region of the gene, then you're pretty much targeting a knockout of the gene. Whereas if you targeted the enhancer, you would still just be controlling its expression rather than destroying the gene. So we're thinking that, you know, instead of, as an alternative to RNAi, for example, we could be targeting the enhancer and controlling gene expression rather than knocking out the gene. I'm hoping that that answers the question. Oh, Emma, you have asked a really long question here. General discussion point, if that's okay, yes. Are any of the SNPs or variants that you and others have identified enhances or intergenic regions shown a degree of pathogenicity as that in key protein coding genes? Yes, to some degree, yes. So, but you might also be aware, I think you know, that a lot of the GWAS hits for diseases such as heart failure are really not landing at these protein coding genes for some reason or other. But troponin, for example, is there. So we are finding SNPs in enhancers of genes such as troponin. Amazes me that most cardiac clinics routinely screen for a panel of genes. Feel that we're missing so much, but don't forget, you know, when these genes are screened for, they're being screened for in monogenic conditions, patients with monogenic diseases, rather than sort of polygenic garden variety heart failure. So I think there is a spectrum. And if you're really looking for monogenic diseases, then these key protein coding genes, sarcomeric genes, are quite often the ones that's the cause for those monogenic Mendelian diseases in high penetrant families. This is saying thank you. So thank you for listening as well. Li Chao, whether are stress response genes or set of genes with similar function more enriched in one TAD and your data? Yes, so actually the location of genes are not entirely classifiable, I feel. So, you know, the stress response genes, the classical ones, MPP and MPPB, are right next to each other. So as we saw, they share enhancers. Actually, MYH6 and MYH7 are also next to each other, and they're also sharing enhancers. We are finding some of these families, but not always are the families situated together. Ritus asked the question, how did you find interaction between enhancers and various genes within or without loop? So this was using, this was using high C and 3C methodologies, chromatin conformation methodologies. And this is something that you can reach out to George. I hope he's listening in. George is the green fingers behind this. So it's basically using anchor and finding out which other locations are interacting with it. That's the principle. And then using sequencing to get to that answer. Lou said, great talk, thank you. Is there any information on new CTCF bounded domains from tag and disease? Do you find any marks that change? Yeah, so we haven't found any of those. It really looks in our hands that CTCF location, CTCF looping is really all quite stable in a tag disease model. Even in our human hearts, we are finding it the same as well. So there's no, at that resolution, there isn't any great boundary changes that we are finding. Rio's asking, is there any data on CTCF or differential? So there's definitely a lot of data on, we have a lot of positive data on K27 acetylation changes in different heart models, HFpEF models, mouse models, and so on. So I think the money is on K27 acetylation changes rather than the looping changes. CTCF level differentially expressed in embryonic and adult heart. I'm afraid we haven't compared that. That's a good question. We might go back and look at that. Yildi is asking, have you studied this gene have a role in oxidative stress response? Yeah, so as said, CTCF doesn't really seem to change in any disease setting. And Rio's asking again, can you differentiate acetylation status in different cell types? Yes. So if you look at the acetylation profile across the genome, it's almost to a stage where you can create these reference maps. And just by looking at the acetylation profile, you probably can say what cell type it is. Or at least which family of cells it is. Is it a fibroblast or endothelial cell and so on? So this is a really good signature for cell types. Jeff Sossman says, thank you for the compliment. Do you know at what development stage these loops are established? Oh, that's a good question. So, no, we haven't looked at developmental stages. So interestingly, so cardiomyocytes are obviously not actively mitotic, but in the mitotic cell systems and other cell systems, when there's mitosis, it's been really very nicely shown how the loops are dissolved, and as soon as the daughter cells come about, the loops are reformed, and they're faithfully reformed. So that's also something really interesting that people are trying to understand a bit more. Next question was, would you know if CTCF function differs during the remodeling period of, so that's not something that we've found. We haven't found any CTCF function changing during the remodeling. So in fact, if we really wanted to be blunt, we'd say that chromatin remodeling isn't occurring even at the CTCF looping level. This is what we're seeing. Whereas the changes really are, as I say, the money's on the acetylation of K27 at enhancer marks. Mukesh is asking, did your study highlight the use of alternative transcription start sites of enhancers for downstream? Yes, so there is quite a bit of that in our data set. And when it gets published, hopefully you can dig into it to look. Did you look at any other histone marks, for example? Yes, we haven't looked at those. So there's been quite a number of data sets generated for histone marks, and they're all published, some to be published. So look forward to a lot of these maps getting put out and people having an integrated study of all of that to understand globally what these histone marks are doing altogether. Julia says, an open-ended question, what are your thoughts on how functional recovery seems to reverse epigenetic changes? What comes first? Oh, that's a really good question. So is it the epigenetic changes that come first, or is it, well, if you talk to an epigenetic-centric person like me, I would probably say that that comes first, but I don't know. I don't have any strong evidence for that. So that
Roger,
would be this.
Is there, just to butt in, Roger, is there an established link between epigenetic changes and functional changes that can be reversed as well? I mean, has anybody really shown that clearly?
Yeah. So the examples of histone deacetylase inhibitors, for example, would be a good one. Although these are deacetylases that can also deacetylate non-histone proteins. So that may not be the best example, but if you take the ones that, on the other hand, inhibit the one that Sap Halder has worked on, which is the BET protein inhibitors. So these are the readers of the acetylation. And if you inhibit the BET proteins, I think he does show that there is some reversal on gene expression. Okay. Which then proves the causality of this.
Okay. Yeah. I'll like to read that.
So Ritu asks, have you looked at other histone modifications? No, not yet, but we will do that. It's something that we should do. It was case 27 acetylation distribution affected by CTCF knockout. So this is a really interesting question, actually. Nice to be the last one, because we did ask the question when you break the loops apart and new interactions appeared. Were these genes now interacting with enhancers that used to already be enhancers, or did new enhancers appear? And it turned out that they were not new enhancers. So all the new interactions occurred with pre-existing enhancers rather than new enhancers. So I think tangentially the answer to this question is that K27 AC distribution was not affected in CTCF knockout. The distribution and existence of K27 acetylation and where enhancers are, they stayed the same. If an enhancer was on, it was on before. And if it wasn't on, it wasn't on still with CTCF knockout. So this again shows the relationship between which comes first.
Okay. Well, thank you. I mean, 28 questions is no small feat. So, so thank you for finding time. I think you definitely deserve to get a good night's sleep after this.
Thank you so much, Davor. It's been good fun. Yeah, this is such an interesting experience.
Many
And
people
well done for coordinating this. This is a really good.
No, I appreciate this, and actually, I look forward to Matt as well from Roger's Lab as well coming and talk soon as well. Roger, thanks once again, and thank you for everybody's attention and for tuning in. And join us again on Thursday and on Friday. And whoever is interested, as I said, we get a lot of support for this from the ISHR, as well as from the Journal of Molecular Cellular Cardiology. And if anybody's interested who is not already a member, hopefully I can convince you to look up some of the details on the society, and here's some of the basic information in this slide. So I will now stop sharing. Roger, thanks once again, and have a good night. Sleep well.
Thank you very much. Good night, everyone.